I'd like to welcome Dr. Bruce Johnson, Professor of Medicine at Harvard Medical School and Dana-Farber Cancer Institute. His talk was Improving Patient Outcomes in Lung Cancer by Targeting Different Driver Mutations. Thanks for joining us.
Well, it's my pleasure to be here.
The somatic driver mutations of EGFR in patients with lung cancer and the clinical response to gefitinib or erlotinib therapy have been recognized. Would you discuss the current research that is attempting to identify other driver mutations?
First of all, to outline what we've discovered with the mutations in EGFR, the epidermal growth factor receptor, have led to changes in our guidelines of therapy. So the discovery took place in 2004, that there was a close association between the response to gefitinib and erlotinib and a rather dramatic response rate of 70 to 80%, and that it worked for about a year, and the survival was 2 or 3 years. And so that the survival of those patients was quite a bit different. This led to a series of randomized trials that were published within the last 2 years showing that patients can about double the length of time their disease is controlled by getting the oral pill gefitinib or erlotinib compared to conventional therapy. Now that success has led us to look for others, and there's an ongoing discovery operation that's taking a look at hundreds of lung cancer samples for thousands of genes for which we can potentially develop targeted treatments.
What is the potential for treating driver mutations, including EML4, ALK, and BRAF with existing targeting agents?
Let me talk a little bit about how we've identified things and the transition we make from the discovery into the treatments. So one of the things that you end up finding out is that once you find that there's a genetic change within the lung cancer, you want to find out if it actually causes the cancer. And there's a series of laboratory experiments that are done both with lung cancer cells as well as in mouse models. And what one looks for is to find out, A, if the gene itself will cause the lung cancer that looks similar to what we see in humans. And the second thing is if you turn it off, either by shutting down the production of the gene or giving something that interferes with the production of the product, that it actually changes the cancer by either causing it to regress or turning it back into a normal tissue. Once you discover that, then you look to see if there's a drug that has the same effect as turning off the gene. So the examples that you make, that you made before, the EML4 ALK had a drug that inhibited ALK. ALK stands for anaplastic lymphoma kinase. And if you turn that gene off, you can kill cancer cells and you make them regress in mouse models that have the activated form of the ALK. So the drug crizotinib ends up causing those things, and it was in phase 1 trials in 2006, and it began entering treatment for lung cancer in 2008 and 2009. And it causes regressions in the majority of patients, and the drug works for about a year, which is about 2 or 3 times longer than chemotherapy. The other one that you mentioned, BRAF. So BRAF is a kinase that's activated most frequently in melanoma, and the research therefore has been focused mainly on melanoma. And in there, the majority of patients who have BRAF mutations get treated with BRAF inhibitors. It causes regression in advanced disease in the majority of times. It's also activated in a subset of colon cancer and in lung cancer, and so we're going to be studying BRAF mutant lung cancer in patients using BRAF inhibitor. However, the thing is, it's only 3% of lung cancer, so it's relatively few. That said, 3% of lung cancer, there's more than 200,000 cases a year, so there's 6,000 people a year that may have the BRAF mutations, and that's a similar number that get advanced melanoma a year.
What are the next steps for research on EGFR, its different mutations, and its susceptibility to inhibitors?
Well, the next steps that probably should be taken is that the EGFR-directed drugs, gefitinib or erlotinib, which are the ones in widespread use in the United States and other parts of the world, were developed against the normal receptor, which we call the wild-type receptor, and that there's a structural change within the receptor that takes place. And so the next steps are probably to develop inhibitors that work better against the epidermal growth factor receptor, meaning that they're specifically designed to inhibit the mutant form rather than the wild-type form. The second thing is that we know that what causes gefitinib or erlotinib to work in the majority of patients. So for example, half of the patients where gefitinib or erlotinib quit working develop something called a T790M mutation or a gatekeeper mutation. And that ends up making it so that the drugs are no longer effective and the cancer will start to grow after it initially worked. And drugs are being developed that work even in that subset. And the hope is that they'll work in the ones where gefitinib or erlotinib have quit working. And also the other hope is that it will actually make them work longer than the original drugs. But there's about a year to 3 years of clinical research before we can make those conclusions.
Well, thank you very much, Dr. Johnson.
Thank you.